Okay, hi. Now we're going to continue the effect of the gene mutation. One of the effect of gene mutation, the gene mutation can lead to the frameshift mutation. What is frameshift mutation? It is a mutation that involves the insertion or deletion of one or more base pairing in the nucleotide sequence of the DNA. When base insertion or base deletion occur, what happens is it will shift or alter the reading frame on the DNA, where it will lead changes to mRNA and thus change the amino acid encoded by the codon, causing the genes to be read in a wrong triplet basis starting from point of mutation. And it will produce the different protein. And this frameshift mutation usually harmful to human, where it causes the major thalassemia. How does this base insertion and base deletion lead to the frameshift mutation? So now we're going to see that this is our normal DNA. So normal DNA, we have AAA, ATG, CTT, and CTC. When the DNA undergo transcription, it will produce the normal mRNA of these bases, and this mRNA undergo translation to produce these 4 amino acids. When base insertion occur, what happens is one base pairing is added to the DNA sequence. So this is the mutant DNA, where from the bases of AAA, ATG, so instead of CTT, we add a new base of cytosine. So cytosine base pairing is added. Cytosine is added to the gene sequence. When the cytosine is added, what happens is it will shift the entire reading on the DNA starting from the addition of the bases. So from the sequence of CTT, when we add another cytosine here, we're going to read the reading sequence on the DNA. Instead of CTT, we get CCT. And this T here will be pushed one basis away from the sequence. So instead of we got the CTC, for the next sequence, the thymine will be pushed to this basis to become TCT. So from the starting of the addition of the base pairing, we will change the entire reading on the DNA, causing the wrong triplet bases will be read. Instead of GAA, we're going to read GGA. And instead of GAG, we're going to read as AGA. So, starting daripada point of mutation, kita tambah cytosine tadi, keseluruhan bacaan pada DNA akan berubah, menyebabkan bacaan pada mRNA berubah. Bila bacaan pada mRNA berubah, dia akan menyebabkan kita menghasilkan wrong codon sequence. So, bila kita hasilkan codon sequence yang berbeza, maka amino acid kita yang terhasil pun berbeza. Sama juga dengan base deletion. When we delete one base pairing ataupun 2 base pairing from the sequence, sebagai contoh, in DNA kita ada AAA, ATG, kita delete this adenine. So, di sini adenine itu being deleted. When the adenine is being deleted, it will shift the entire reading on this DNA. Dia akan tukarkan keseluruhan bacaan pada DNA sequence starting daripada base yang kita delete tadi. So, instead of kita dapat ATG pada second reading, disebabkan kita buang adenine tadi, instead of kita dapat ATG, this cytosine akan bergerak untuk lengkapkan triplet bases. So, kita akan baca TGC instead of ATG. Kita baca TTC instead of CTT. And kita baca TCC instead of CTT. Okay, so base deletion and base insertion will cause the entire reading on DNA change. So it will change the entire reading on mRNA starting from the point of mutation. Starting daripada mutation berlaku sahaja dia akan berubah bacaan. Bila kodon tersebut salah dibaca, kodon tersebut salah dibaca, dia akan menyebabkan keseluruhan penghasilan amino acid berubah. So dia akan menghasilkan different amino acid. So bila different amino acid, maka kita akan menghasilkan different protein. Okay, so this effect is very harmful in gene mutation. So gene mutation, the types of gene mutation ada 4: base substitution, base inversion, base deletion, and base insertion. Base substitution and base inversion could lead to the missense mutation, nonsense mutation, and silent mutation, while base insertion and base deletion akan menyebabkan frameshift mutation. So between missense mutation, nonsense mutation, and silent mutation, frameshift mutation is the most harmful one. Kenapa dia harmful? Sebab dia akan menyebabkan keseluruhan bacaan pada base sequence kita berubah sepenuhnya, starting daripada point of mutation. Okay, itu sahaja yang kita belajar dekat dalam gene mutation.